Hello students, continue with the chapter The Fundamental Unit of Life. Today we are going to discuss some of the cell organelles which perform important functions in cell. So the first cell organelle is cell membrane. Cell membrane, which is also known as plasma membrane or plasma lemma, it is actually the limiting boundary of each cell which separate cytoplasm from its surrounding. It is actually the barricade, or it provides it separates the cell from its surrounding. Outermost covering, of course, so protection is there. Now, the widely accepted model of cell membrane was given by Singer and Nicholson in 1972, and he described the model, trilaminar model, where trilaminar means the cell membrane is actually made up of 3 layers, and where the 2 layers of lipids, which are actually most probably the phospholipids, are there. So 2 layers of phospholipids, and between the 2 layers of phospholipids there is a protein layer. So actually the protein layer is sandwiched between 2 layers of lipid. This one is outer lipid layer and this is inner lipid layer. Okay, now because here is the cytoplasm, this is the inner side of the cell and here is the outer side. Okay, so 2 lipid layer, this one is the first layer and this one is the second. Let me draw 2 molecules of phospholipid. That is, one phospholipid is this way and the another phospholipid is this way. Each phospholipid has 2 parts, head and tail. The tail is actually the fatty acid tail. Fatty acid tail. So this is the tail of the upper phospholipid and these are the tails of lower lipid layer, and this one is the head. So head is actually the glycerol molecule which is attached with the phosphate and choline. That is why it is phospholipid. Okay, so tail is of fatty acids. The tail is also head. So the head is hydrophilic end and the tail is known as hydrophobic end. So this is hydrophobic, which is actually non-polar, and hydrophilic means polar end. Phospholipid bilayer. In between these 2 layers are the proteins present, and the proteins which are present, they are of different type. Like they may be broadly categorized into 2, extrinsic and intrinsic, or this is written here, integral protein, which is actually present towards the inner side. It may be, it may not be, present throughout the cell membrane. It may be like this much only, but deeply inside the cell membrane. So integral protein, and which is also known as intrinsic protein. Intrinsic protein, and the outer protein which is known as extrinsic protein. Now, extrinsic protein means peripheral protein. Integral protein means intrinsic protein. So, these peripheral protein can be removed easily, but the integral protein or the intrinsic proteins cannot be removed easily. Some of the intrinsic protein, they provide even the channel for the pass, for the movement of the substances from outside to inside. Because the cell membrane does not allow all the material to pass through it, it will allow only some of the material, and that is why the cell membrane is also known as selectively permeable membrane. Well, this is the model which is widely accepted, and one more important feature of this model is that this model is actually quasi-fluid structure, or this cell membrane is actually dynamic and it has got fluidity. Now Fluidity means it is flexible, and the proteins as well as lipids, they show certain types of movements like lateral movement. This way, it can change the shape, like change the position. So lateral movement is there because the movement which is present in the protein as well as lipid, it provides the fluidity or the flexibility in the cell membrane. So it is dynamic also, it is not static. Cell membrane is not static. So this model is widely accepted because it has the two important features. It shows the two important features. Let me write, it is not static. Two features of cell membrane. First one, trilaminar. Two phospholipid, that is phosphobilipid layer along with proteins. The second one, it has quasi-fluid structure, or the cell membrane has fluidity. Okay. Yeah. Cell membrane is known as selectively permeable. See, certain types of membranes are present. Let me tell you different types of membranes. The first one is impermeable. The second type is permeable. Third is semi-permeable. And the fourth one is selectively permeable. Selectively permeable. Let me explain you one by one. Impermeable, which does not allow it will not allow the solute or solvent. It will not allow any of the substance, not the solute, not the solvent. Example of impermeable membrane, if the cell wall has the suberin or lignin deposition, then that membrane becomes impermeable. Permeable membrane, which allow both solute as well as solvent. Everything can be, inside the cell. Okay. And the example of permeable membrane, primary cell wall, which is made up of cellulose. Semi-permeable. It allows only solvent, not the solute. So semi-permeable, it is actually egg membrane and parchment membrane and the gallbladder membrane. These are some examples of semi-permeable membrane. Now in many diagrams, or in, you can see in many books, this is written that the plasma membrane is semi-permeable. The plasma membrane is actually not semi-permeable. The plasma membrane is selectively permeable. So it's written here selectively permeable. Why? Because it allows or permits the entry and exit of selected material. Selected material means selected solute and selected solvent only. Selected solute, selected solvent. What does it mean? Those material who are beneficial for the cell, maybe solvent, maybe solute, cell membrane will allow. And any solute or solvent which is harmful for the cell, it will not allow. So it will allow selected solute and selected solvent. Selected solute as well as solvent. So the cell membrane and the cell organelles membrane are selectively permeable. Right? Now let's come to the functions. It regulates, of course, the movement of molecules inside and outside. This now we have seen it is selectively permeable. Second one, maintaining the distinct shape of the cell, because it is the boundary which separate the cytoplasm or the cell from the surrounding medium. It acts as a mechanical barrier. It does not allow any material, it does not allow anything to enter, right? So mechanical barrier. Cell membrane gets modified, yes, according to the function. For example, like intestine, where the cell membrane invaginates and form the microvilli-like structure so that it can increase the surface area, and this surface area, we can see here that the area of the straight line and the area of this coiled finger-like projections, the surface area is more here. So more surface area, more absorption. So for efficiency of its function, right, it is modified into different types of structures. You can see the example of amoeba, where the cell membrane form the pseudopodia to engulf the food, to engulf the water droplets, pinocytosis and phagocytosis. So cell membranes get modified according to the function. Okay. Now we come to the important function, that is transport of material from cell membrane. Now we will see the different important two processes here we will study. The first one is diffusion. This term you can feel so many number of times in a day in your daily life, like when you light the incense stick in the pujagar, then you can smell that fragrance in your kitchen, in your study room. Why? Because that fragrance will come from pujagar to your study room, because the diffusion is what it allows the molecules to move from their higher concentration to their lower concentration. So where the higher concentration is present, molecules will start moving on their own without using energy. No energy required. No energy required. And that is why it is known as passive process. Okay. So no energy required, and the molecules will move from higher concentration to lower concentration. So how will you define diffusion? The net movement of particles from area of high concentration to lower concentration. In another way, you can write that along the concentration gradient. Along the concentration gradient. Not against, but along. From high to low. Example, if you can see that when your mama add food color, then there is no vigorous mixing and all. Just stir it once or twice. This food color can change the color of the complete food. Okay, so high concentration of color moves to the lower concentration. In living organisms, we can see the example in plants as well as the animal, which is given in your NCERT. So these examples are important one because these are from NCERT. First one, in plants, exchange of gases in respiration, exchange of gases in photosynthesis, and release of water in transpiration. All these three processes involved diffusion. How? Because in diffusion, the water vapor, first one, transpiration we are taking. Water vapor is released in the atmosphere through stomata. Why? Because the stomata has the water droplet and outer atmosphere is dry. So less amount of water is less here and water is more. Water is less in the environment and water is more in the guard cell. So the water will be released through the osmosis process. Okay, guard cell, it is more. Now, second process, photosynthesis, where the CO2 is taken in to perform photosynthesis, and the oxygen which is formed, a byproduct, this oxygen will be released. Because oxygen is formed inside, so more O2, right? So it will move out. More CO2 outside, so CO2 will enter. So exchange of gases along their concentration gradient in photosynthesis, same in respiration also. In respiration also, CO2 which is formed, it will be released, and the oxygen which we require, it will be taken in. So exchange of oxygen and carbon dioxide in lungs and also in the tissues. Because when the arterial blood carries the oxygen in oxygenated blood to the tissues, in tissues the CO2 will be, in tissues the O2 will be released, and in the blood the CO2 will be taken. From blood oxygen is delivered and from tissues CO2 is taken. So exchange of gases along the concentration gradient. Okay. Now the next mode of transport, that is osmosis. The second process is osmosis, which is equally important. That is the movement of substances from the area where more water concentration is there to the area where less water concentration is there. See, we have just now studied about the diffusion. In diffusion, we have discussed about the movement of gaseous molecule. So diffusion is actually generally we use for the movement of gaseous molecule. Movement of gaseous molecule. From higher, of course, to low concentration. But here in osmosis, we actually see the net movement of water molecule through the cell membrane from area to, of course, high to low. It's written here water potential. Water potential means where the free energy of water molecule is more. Free energy is more. So potential energy, so more water, more free energy. So from this way also we can say that the water will move from higher concentration to lower concentration through semi-permeable membrane. That is the complete definition. Now it's in this picture you can see that here is a concentrated. You can see here concentrated sugar solution. What does it mean? Here the sugar molecule is more and the water molecule is less. Blue is water molecule and the purple is sugar molecule. So more amount of sugar. So concentrated sugar solution. Dilute sugar solution. Sugar is dilute. Sugar is less. Then more water. So water is more. Here water is less. Now you can easily find out, make out that the water will move from its higher concentration to lower concentration. So here is dilute, here is concentrated solution. Now where the water is less, water will move. When water will move here, then automatically there is a rise in the water level. So this rise in the water level, it is due to more water is drawn into the concentrated solution. Till this process continues till the concentration on both the side will be same. Both the side will be same. What does it mean? There are different types of solutions depending on the amount of water, that is amount of solvent or amount of solute. We will see in the next slide. Types of solutions. Types of solution on the basis of amount of solute and amount of solvent. Actually the solvent is the medium where the solute is dissolved. Okay. Here generally we take the solvent as a water. So solvent is water and solute are the particles which we dissolve in that solvent. Okay. Now look here the concentration. 20% solute and 80% water inside the cell. Okay. 20% solute, 80% water. Okay. Outside. What does it mean? Equal concentration of both inside and outside. So a solution where the same concentration of solute and water, solute and solvent outside and inside. Then the medium, actually the medium has the same concentration as the cell. This type of solution is known as isotonic, where the movement of water molecule is going out also, water molecule is coming in also. There is net movement is zero, because equal amount of water is moving out, equal amount of water is moving in, because at both the sides, in both the sides, the concentration is same. So there is no net movement and the overall concentration on both the sides remain constant. Then there will be no change in the size of that, shape of that cell. There is no change. Okay. So first type we have seen isotonic, concentration same outside inside. Hypertonic, hyper, more. The solution which has higher solute concentration than the another cell or another solution. One solution has higher solute. When the solute is higher, then less water. Of course, less water and high solute. Water particles will move out of the cell. What does it mean? Here is the cell. Here is the solution or medium. Now look, in cell 20% solute, 80% water. What does it mean? More water inside. But in medium, okay, in medium 40% solute and 60% water. So less water. In medium, less water. In cell, more water. What will happen? Water will move from higher concentration to lower concentration. Water will move out. That process is known as exosmosis. Why exosmosis? X, outside. Water is moving out of the cell, and you can see here the size of the cell, decreases. When we keep the cell in the hypertonic solution, hypertonic medium, then the size will decrease because exosmosis occurs, because outside the water is less. So the amount of water in cell is more. Of course, water will move from its higher concentration to lower concentration, so size will decrease. Look at this type of solution now. Hypotonic, where the case is opposite. What? Now, 20% solute and 80% water. Here 10% solute only. Here 20% solute. Here 90% water. Here only 80% water. What does it mean? More water. Here less water. Water will move from its higher concentration to lower concentration. Water will enter inside. The solution or the medium where more water is present than the cell, this type of solution is hypotonic. Cell increases in size and osmosis occur. And osmosis means osmosis occurs, that is water is moving inside the cell. Now in your NCERT, we have seen now the three types of solution: isotonic, hypertonic, hypotonic. Please remember, because 3 mark question generally come on this topic, on this particular topic, that if the cell is kept in hypertonic medium, what will happen to its size? If the cell is kept in hypertonic medium, what will happen to the size of the cell when it is kept in isotonic medium? So you should first know the concept of isotonic, hypertonic and hypotonic medium or solution, so that you will be able to give the answer. There is an activity of egg in your NCERT. So we will see the activity. Be careful, attentive, because the egg is actually, we all know that the egg is a shelled structure, and this shell is actually calcium carbonate. inside that shell is the membrane, egg membrane is there, and the egg membrane will absorb water when it is dipped in hypotonic medium, and it will lose water when it is dipped in hypertonic medium. But before that, we have to remove that calcium carbonate, and in this activity you will see that first the calcium carbonate shell is removed by using the acid, and then this end osmosis and X osmosis activity you can see in the.
Dissolve the outer shell by placing an egg in vinegar. Vinegar has a chemical called acetic acid. Eggshells contain calcium carbonate. When calcium carbonate in the eggshell comes in contact with acetic acid, a chemical reaction occurs. Calcium carbonate will break down to form carbon dioxide, water, and calcium acetate, thus dissolving the calcium carbonate shell. Within seconds, you'll see bubbles form. These bubbles are carbon dioxide that are produced when acetic acid breaks down the eggshell. Within 3 days, you should have a semi-permeable membrane free of the hard outer shell to conduct your experiment. Before starting our experiment, we will first mass our egg using a scale. Egg A has a mass of 94 grams. Egg B has a mass of 103 grams. Egg A we will place in a hypotonic solution, water. Egg B we will place in a hypertonic solution, thick and sugary corn syrup. Egg A now has a mass of 97 grams. Egg A increased slightly to 103% of its original weight. We can conclude when a semi-permeable cell is in a hypotonic solution, water will enter the cell. Egg B now has a mass of 59 grams. Egg B decreased to 57% of its original weight. We can definitely conclude that when a cell is in a hypertonic solution, water will leave the cell. In fact, this is evident.
Now, you have seen that video that egg, when it is kept in hypotonic medium, it increases in size, and when the cap is dipped in hypertonic medium, then its size decreases. Now, here is a question for you. When the cell is exposed to hypotonic conditions, there is a net water movement into the cell. True or false? See, the question is cell exposed hypotonic condition. So, here is the beaker where the water is there. Okay, the solution is there. Now, I keep the cell. Suppose this cell is raisin. We all eat raisin, right? So, when we keep the raisin for 1 or 2 hours in a hypotonic medium. Hypotonic. Now we will see the meaning. Hypotonic means more water, of course. And in raisin, less water. So what will happen to the size? Of course, because less water and more water. So water will move inside, and after some time, when we see what will happen to the size will be increased. This raisin will increase in size due to end osmosis. So the answer is yes, true. Okay, so the answer is true. Now, the next question. The next question is, hypotonic solution means more solute and less water. More solute and less water. True or false? Hypotonic medium, just now we have seen in the previous question that hypotonic means more water. But more water less solute. So hypotonic solution, more solute and less water, true or false? It is false, because opposite is correct. Hypotonic medium, where the more water concentration is there, solute is less. So the answer is false, right? Now, identify the type of solutions A and B. Observe the picture and see. You can easily find the answer by just observing these slides, these pictures. How? Because look here, this is water, of course, and here is the cell. You can see here more water is going in and less water is coming out. More water is entering. The size of the cell is larger here. You can see in B, right? In B, less is entry is less, but release, outside movement of water is more. The size of the cell is, shrink. Now look at the third. Equal amount of water is entering, equal amount of water is coming out. The size of the cell is the medium, that is remain. So you can easily see the answer. This must be hypotonic, this must be hypertonic, this must be isotonic. So let's see the answer. Yes, hypotonic, A, B is hypertonic, and C is isotonic. Now, important function of cell membrane. One more important function. In the previous slide where we have seen the important functions of cell membrane, the last function I explained to you that the cell membrane can change the shape to get the food and water and also to increase their surface area. Okay, this is very good example that amoeba is single celled, it is unicellular, and it eats by endocytosis and it releases waste by exocytosis. Amoeba itself is, capable of performing all the vital functions, right? Because of the presence of this cell membrane which can change its shape. So, by pseudopodia, you can see it easily engulfs the food. Endocytosis means inner to the cell. So, the endocytosis means the food particle is engulfed. It is amoeba is taking food particle inside. The food vacuole is formed here. Then the digestion will occur. Then the digested food will be, of course, absorbed by the cytoplasm, and the waste material which is left in that will be converted into contractile vacuole, and this contractile vacuole will releases excretory matter into the water only, because amoeba is an aquatic organism. So by exo means outside the cell. So of course, these two, that is exocytosis and endocytosis is the function of cell membrane, and it requires energy. Very, very important. It requires energy. Children, in the previous two processes which we have seen, diffusion and osmosis, because the movement was along the concentration gradient and no energy was required. So these two processes, osmosis and diffusion, we have seen they were passive. No energy required. So passive. But endocytosis and exocytosis, active, because it requires energy. Now you can see in this slide.
Within a unicellular organism are carried out by the single cell itself.
So endocytosis and exocytosis.
In amoeba, a single cell is responsible for movement, intake of food, exchange of gases, and excretion.
Now, we'll come to the next cell organelle, that is cell wall. The cell wall is the outermost boundary which is present in plant cell. Because cell wall is absent in animal cell. Cell wall is present in plant, cell wall is present in fungi, cell wall is present in bacteria. Okay, bacteria is prokaryotic. We'll see prokaryotic and eukaryotic organisms and their differences in the next coming slides. Okay, cell wall is the outermost layer which is present in plant cell, fungus, and eukaryotic plant cell, fungus, prokaryotic bacteria. Okay, but it is altogether absent in animals. The primary cell wall and the secondary cell wall, different layers. The first laid cell wall is primary, and inner to the primary, mind it, inner to the primary will be secondary, tertiary, like cell wall. So primary cell wall has the key material in it is cellulose, hemicellulose, and pectin. And these materials are of course dead. So the cell wall is dead. It is not living. Okay. So cell wall is dead. Cell wall is dead. And we have seen that this is actually permeable membrane. Primary cell wall is permeable. Major function of cell wall is actually act as a pressure vessel which prevent over expansion in plants and the fungus and the bacteria. Why? It actually prevent the plant cell and the and the organisms in which cell wall is present from bursting, because when they are present in aquatic medium, then the aquatic medium, outside medium is hypotonic. So the cell tends to absorb water by end osmosis, and if water will enter continue, then the cell may burst. But actually what happens? When the cell absorbs water, cytoplasm, inside that cytoplasm, the water level will increase, and that creates a pressure on the cell membrane so that the size of the cell will increase. Of course, if water will enter, then the size will increase because this cytoplasm exert a pressure. Actually, what is the role of cell wall? The cell wall then exert pressure inside. Okay, so cytoplasm is exerting pressure on outside, but cell wall is, exerting pressure to the opposite of the cytoplasm. And when this wall pressure, cell wall, which is the pressure which is exerted by the cell wall is known as wall pressure. When the wall pressure becomes equal to the turgor pressure, then this osmosis stops. And when osmosis stops, there will be no movement, no increase in the cell size. This way, the cell wall will prevent, actually protect the cell, plant cell from bursting. Okay. And of course, because it is the outermost layer, so protection of the cell is the important function, maintains the shape of the cell, maintains, rigidity, it's provide rigidity. It is not very flexible, so it is rigidity. Maintains the osmotic integrity or the turgidity of the cell. Now, this is the important term, that is plasmolysis. Plasmolysis means the lysis of plasma membrane. What does it mean? When the cell is kept in hypertonic medium. Look at this condition. One cell is there, right? And the medium is hypertonic. What does it mean? Less water. Hypertonic medium means less water. So inside the cell, more water. And we all know that in osmosis, the water will move from its higher concentration to lower concentration. So when a cell is kept in hypertonic medium, then cell tends to lose water. Now, the cell loses water, it will shrink. But what actually happens, the cell wall which is present, this cell wall is rigid. The cell membrane is flexible. So when the cell releases water, it shrinks, but cell wall does not shrink. Only the cell membrane. The cell membrane starts, shrinking, right? And it will move away from the cell wall, initially from the corners. So cells showing contraction of cytoplasm along with the cell membrane. Because the cell is continuously losing water. So cytoplasm shrink, and along with the cytoplasm, the cell membrane also shrink. But the cell wall being rigid, it remains unaffected. Okay. Now slowly the cytoplasm and the cell membrane along, this is actually the vacuole which is present in vacuole, because we all know that this large central vacuole which is present in the plant cell, and this one is the nucleus. So the vacuole slowly, it is becomes smaller in size, right? So the shrinking continues, and the plasmolyzed cell you can see in the last diagram. And what actually is filled in the space between the cell membrane and the cell wall? This is the cell wall, this is the cell membrane, and here is the cytoplasm. So what actually is filled here? Here the filling is of hypertonic solution which is present outside the medium. Hypertonic solution. This is very important, children, because it is generally asked that the gap between the cell membrane and the cell wall is filled with what? With water or with gas, that is the oxygen, CO2, what? So this gap is filled by the hypertonic solution which is actually present outside. So this is the important term, plasmolysis. Now again, a question for you. Dash prevents a plant cell from bursting when kept in hypotonic solution. See, the plant cell, it is provided with rigid cell wall. And the rigid cell wall protect not only plant, but the rigid cell wall present in fungi, the rigid cell wall which is present in prokaryote, that is bacteria, cyanobacteria, prokaryotes. It protects cell from bursting because when the cell absorbs water, right, it will increase in size. And if the cell is kept in hypotonic medium, then the water will continuously enter inside that. So the cell will increase in size continuously and automatically it will burst. But actually what happens? Because of the presence of cell wall, the cell wall protects it from bursting. How? When the cell absorbs water, its cytoplasm exert a pressure on the cell membrane so that the size will increase. And what actually happens? The cell wall exerts pressure in opposite direction. And when this pressure which is exerted by the cell wall, it is known as wall pressure. And the pressure which is exerted by cytoplasm, it is actually turgor pressure. So when the wall pressure becomes equal to the turgor pressure, osmosis stops. And this way it will easily protect cell from bursting. So the answer will be cell wall. Okay. Is plant cell wall is living or dead? Question number 2. Cell wall, the important constituent of cell wall is cellulose. Of course, in the primary cell wall. And in the secondary cell wall, then the suberin or lignin or all these, deposition, all these chemicals will be deposited. The primary cell wall is, of course, the dead. And in normal plant, the primary cell wall is present. So cell wall is dead and it is permeable. It will allow solvent as well as solute both to enter. So cell wall is actually dead. Children, thank you. That's all. We have studied about the cell membrane, the function of the cell membrane, where we have studied the diffusion and osmosis, and then we have studied the structure of the cell wall and the function of the cell wall, and at the end we have studied just now the plasmolysis. Thank you. In the coming video, I'll explain you the function, the structure and the peculiar feature of prokaryotic and eukaryotic cell. Thank you.